Hello, my name is Sophie, and today I'll be presenting on ensemble learning algorithms to predict scaffold matrix attachment regions, or SMARs. SMARs are genomic elements that anchor DNA to the nuclear matrix. This anchoring facilitates the structural organization of chromatin into functional domains and plays a critical role in gene regulation and DNA replication. Mutation in these regions have been implicated in metastatic cancers, which highlights their biomedical significance. Although some DNA motifs and sequence patterns associated with SMARs have been identified, there is currently no comprehensive database of SMARs across the human genome. An accurate identification of SMART sequences could be a valuable resource for scientists studying cancer and other diseases. A robust and reliable method for SMART detection would also enhance our understanding of the regulatory functions and potential therapeutic implications. Currently, the methods to predict SMARs are insufficient. Our project aims to fill that gap by using machine learning models to detect SMARs accurately and consistently, an advancement that could enhance cancer research and deepen our understanding of genome organization. On the right, we can see a diagram of SMARs in the nuclear matrix. Introduction. To understand the importance of this project, let's first look at what SMARs are and why they matter. So, scaffold matrix attachment regions, or SMARs, are segments of the human genome that anchor chromatin to the nuclear scaffold or matrix. This structural connection is important because it helps organize the genome in three dimensions and plays a vital role in regulating gene expression, as shown in the image on the lower right. The nuclear matrix is a three-dimensional framework within the nucleus. It supports many nuclear processes and has been observed interacting with SMARs under a variety of conditions, as highlighted by Narwade et al. 2019. Importantly, SMARs are also known as hotspots for retroviral integration. This is the mechanism by which retroviruses, such as HIV, insert their genetic materials into the host organism. This makes them relevant in gene regulation, but also in virology and gene therapy. SMARs are also been shown to influence epithelial mesenchymal transition, or EMT, which is the process in which epithelial cells acquire migratory and stem cell-like processes. This is a crucial step in malignant progression and the spread of cancers, including breast and pancreatic cancer, as shown by Adhikari et al. 2014 and Dongre et al. 2019. Despite their biological importance, most current methods for identifying SMARs are unreliable. Such as Evans et al. Report that prediction accuracy remains low across various computational tools. This inconsistency detection is a key gap that our project aims to address using modern machine learning techniques such as ensemble learning. So, to approach this challenge, we implemented and optimized multiple machine learning models to classify DNA sequences as SMAR or not SMAR. So, the core key models we trained are random forest, k-nearest neighbors, XGBoost, and neural networks. Each model was trained on 435 experimentally validated SMARs from the HeLa cell line using data provided by the ENCODE project. For the negative control group, we used inter-SMAR sequences, that is, segments between the confirmed SMARs that were experimentally proven. To enhance model accuracy, we emphasized biologically relevant data and biologically relevant features such as measuring the AT richness. For data collection, we began by collecting 435 SMARs from the ENCODE dataset. This only gave us a start and end position, so from there we used Google Apps script integrated with Google Sheets and the UCSC genome browser to extract the DNA sequences corresponding to those regions. To balance this dataset, we used SMOTE, which is the synthetic minority oversampling technique, also scrambled DNA of known SMARs to generate negative examples. This produced a final distribution of around 1,500 non-SMARs and around 500 SMARs. A custom pipeline was then built to process the sequence and search for biologically important motifs, such as AT richness, ATTA motif, and 6W repeats, where W is A or T. As previous literature has shown, these motifs were associated with scaffold matrix attachment regions. Finally, we split the data in an 80-20 train test ratio. So the first model we built was a random forest model. This was chosen for its robustness and ease of interpretation. We then had developed additional models, including the k-nearest neighbors and the XGBoost. We're also in the process of developing a neural network and eventually a naive Bayes model. The end goal is to use bagging, which is an ensemble learning technique in which you combine multiple models to improve predictive accuracy by reducing overfitting and leveraging the individual strengths of each model. So this will be done by combining our base learners, which are k-nearest neighbors, XGBoost, random forest, neural network, and naive Bayes. So here on the next slide shows our preliminary results. Random forest achieved the highest accuracy, being an 87.5% accuracy. Next, XGBoost received a 81.6% accuracy. K nearest neighbors has an accuracy of 67.9. On the bottom left, we can see an ROC curve. This indicates how accurate the model is by showing how close it is to the upper left corner. As shown here, a random forest model and K nearest neighbors model are pretty accurate. These results indicate that our approaches and well-tuned hyperparameters have the potential to significantly improve classification for smart prediction. So this project offers several valuable takeaways. The first being that we achieved a relatively low error rate in classifying SMARs. We compile a list of DNA motifs that serve as reliable indicators of SMAR presence, such as AT richness, ATTA, and 6As or 6Ts. Furthermore, since SMARs are known to correlate with retroviral integration hotspots, our work may aid in the identification of these hotspots. And finally, our method could help researchers better understand the molecular basis of cancer, potentially improving clinical outcomes for patients with SMAR-related mutations, such as in breast and pancreatic cancers. However, there are a few limitations, some of that being we still need more data, especially biologically relevant negative examples. Furthermore, some of our models are prone to overfitting, especially when they're trained on augmented data, such as from the SMOTE technique. And in the future, as we expand our data set, computational and processing power may become a concern. Moving forward, we plan to finalize our ensemble model using bagging techniques with random forest, XGBoost, K-nearest neighbors, and naive Bayes. We also plan to test the input of multiple data inputs such as SMOTE or biological data, determine which one is most accurate. And finally, we plan to apply the model to genome-wide identification of novel SMAR loci. We're also working on making our model into an R package, making it more accessible to scientific researchers. So by identifying these SMARs using our model, we can help researchers uncover new insight into chromosome organization and metastatic processes. This will be important for improving diagnostics and treatment planning for SMAR mutation-associated cancers. Furthermore, identification of SMARs provides a tool for tracking retroviral integration, which could be relevant for gene therapy and virology research. Here are our sources, and I'd like to thank my principal investigator, Mr. Kuna, for his guidance. I'd also like to thank ASDRP and the Olive Child Foundation for this incredible opportunity, and the ENCODE project and UCSC for the tool that allowed us to obtain this data. So thank you so much for listening, and I'd be happy to take any questions.